Hello, everybody. My name is Chris Griffiths. I have the privilege of being the president of the ESDR for the current year. And I'd like to welcome you to an initiative on behalf of the ESDR. The chairman of this particular initiative is Professor Ellie Sprecher. And this is where we're going to discuss scientific and educational topics live with you over a series of webinar programs. And the reason that we're doing this, as most of you will be aware, is that we decided, rightly so, 2 or 3 months ago, to cancel our annual ESDR meeting, which was to be held in Amsterdam this year, and move the 50th meeting, as it would have been, into 2021. But we wanted to maintain contact with you and provide education and scientific discourse. We've called this the ESDR Kitchen. It's going to be an eclectic mix of cooking. It's going to involve 4 main programs: Sweet and Sour, which is 2 dermatologists or skin biologists debating an important, contentious scientific topic relevant to dermatology. Freshly Baked, there's going to be 3 quick-fire presentations on high-impact, recently published research from the researchers themselves. And then Molecular Cuisine, where an eminent dermatologist or skin biologist is going to discuss his or her pathways to discovery over the course of their career. But we're going to start, as all cooking does start, with the recipe book. And this is where we're going to discuss and explain research techniques. I'm going to now hand over to 2 of my ESDR board colleagues, Enikö Sonkoly and Julien Seneschal, who are going to take you through this session, which is going to be presented by Tiago Matos. I'll come back to you at the end of the program to let you know what we're going to be doing next time. Enjoy it.
Hi, thank you, Chris. Hi, everybody. Welcome in our ESDR kitchen. So I'm Julien Seneschal, professor of dermatology in Bordeaux, and I will have the chance to moderate this first session. And I will share the moderation with Enikö Sonkoly, and I'll now let her introduce our first speaker.
Yes, good afternoon, everybody. Hello. My name is Enikö Sonkoly. Thank you for the introduction. And I'm at the Karolinska Institute, Stockholm, Sweden. I have the pleasure to moderate this session together with Julien. This is the very first event within the ESDR Kitchen online seminar series. Well, cooking starts with a recipe. So today we are going to start the series with the recipe of mass cytometry, which will be presented by Tiago Matos from the Amsterdam University Medical Center. Tiago Matos works as a research fellow there and also as a clinical dermatologist. Some of us had the pleasure to listen to him last year at the ESDR meeting. He has a long-standing interest in cellular immunology, in particular skin-resident T cells, but today what he will do, he is going to make mass cytometry simple for us. I'm very sure that you will have many questions, and I would really like to encourage you to make this interactive, to ask questions. You can ask your questions in the Q&A section. Please just write your questions there. You can write them anytime during the presentation, and Tiago will answer the questions at the end. So please, Tiago, welcome to share your screen and start your presentation.
Thank you very much. And hello, everyone. Thank you for joining us. And I would like to start by thanking the ESDR for this incredible opportunity to give the first talk of the new ESDR Kitchen series. My name is Tiago Matos, and I'm a resident in dermatology and a research fellow in Amsterdam, the Netherlands. In this talk, we will be discussing mass cytometry as a novel tool to decode the complexity of the biological systems. Guided just through a brief overview, and we look to the introduction about mass cytometry, steps of mass cytometry experiment and the protocol design. We'll use a study of the regulatory T cells as an example of that design that is required, the analysis strategies and tools that we can use for the complex data that we get, and key research studies that have been done within our field. And I will leave you with further studying afterwards. And this presentation is really based on 2 articles that we wrote for the Research Techniques Made Simple series of the JID, the Journal of Our Society, which I would strongly recommend and advise to any of you to check out the articles of this series and even consider to submit an article to this series. It was a very enriching opportunity to me to write these articles, especially because I got the chance to work directly with the editors of the journal, just like Jody Johnson, get figures done by the journal illustrators, and these articles even include a presentation and a quiz that can be used by anyone, for example, during a journal club or even at lab meetings. And mass cytometry is the new alternative to the conventional cytometry, flow cytometry, that uses fluorochromes attached to the antibodies. Routine analysis has limited to usually 20 parameters that can be analyzed simultaneously. And its limitations are the wavelengths of the fluorochromes that can overlap. It requires then a compensation that introduces noise. And we can also not forget the fact that the cellular structures also have a natural fluorescence. While in flow cytometry, we use a fluorochrome. In mass cytometry, we use a stable metal isotope conjugated to the antibody. And there are more than 50 isotopes that we can use simultaneously. Some of the advantages are that these are stable metals leading to a detectable single peak. It doesn't require any compensation since there's really little overlap. And we see here the metals that are used. In pink, the metals isotopes that we can use to conjugate to the antibodies of interest. Why these metals? Well, they have the advantage of being absent from the biological structures. They are also not radioactive, and they are heavier than any of the metals that we found in the biology, but still heavy enough to be detectable by our cytometer. Then in dark gray, we see the metals that we can use to identification of the cells. The iridium will intercalate within the DNA. So any cell having a nucleus will allow the intercalation of iridium, which allow us then during the analysis to identify single cell events. And we'll use rhodium and platinum as to identify dead cells. And CyTOF technology may lead to a greater comprehension of the cutaneous cellular phenotype heterogeneity, the development, the hierarchy, and the relationship to other tissues, especially because it allows an integrated and simultaneous study of different cell states, such as proliferation, hypoxia, enzymatic activity, and also the deeper understanding of expression of mRNA, transcripts, cytokines, growth factors, and many others in each specific subset. Here we see a simplified mass cytometry experiment that starts with a single cell suspension, either from blood or tissues. Of course, after the tissue digestion, dead cell removal, or cell sorting. The metal labeled antibodies that are incubated with the cells, we can use 35 or more antibodies simultaneously, and then the cells are injected in the cytometer. In this figure, we see the mechanism of mass cytometry, also known as CyTOF, that stands for cytometry by time of flight, TOF. And when introducing the cells in the cytometer, The cells are they go through the nebulizer, which will produce the stream of single cells. The cells are individually atomized and ionized as they pass through the inductively coupled plasma torch with temperatures above the 7,500 degrees Celsius. The low mass elements such as carbon and nitrogen will be filtered out by radio frequency quadruples before entering then the time of flight chamber, or also known as TOF chamber. Then we are left with the metals that we use to tag the different isotopes to the cells, where they are separated by their mass to charge ratio. So the lightest masses arrive first to the detector and the heaviest the last. And from each cell that is introduced, then we obtain a so-called fingerprint of mass spectrum. So here you see in the bottom of the slide, 3 different color peaks correspond to 3 different masses that correspond to different probes that we introduced coupled to the antibody to the cells during staining. It will then be integrated into an FCS file that we can later run and analyze the data. You see here also that the limitations of this technique is the fact that the cells are destroyed, they cannot be recovered, and there are no scatter profile and no sorting. In mass cytometry, it is imperative to establish meticulous experimental strategies and to have clear objectives at the onset of each experiment, since this will strongly influence the results that we'll obtain. After defining the specific experiment aims, it's important to define the type of cells that we want to use, the experimental conditions, the comparative groups, and which controls. In some cases, facts of magnetic activated cell sorting can be used to enrich for rare subsets to avoid the long acquisition time of CyTOF. Then we list the markers necessary to define each cell population. We can use up to 50 different markers. We select the metal isotopes that we want to conjugate to each marker, each antibody, and we developed the metal antibody panel. These panels we can use either commercially available panels, such as this one that includes 30 different markers of antibodies provided by Fluidigm, which allows the detection of 37 immune cell subsets simultaneously. And we have already over 650 metal conjugated antibodies that are commercially available. Nevertheless, if a certain combination is not available, it is possible to self-conjugate one of the existing 42 metal isotopes to the antibody of our interest. But then we need to proceed, of course, with validation and titration. Finally, we need to test the staining protocol, the compatibility, specifically when we use different markers, surfers of intercellular, internuclear, or tetramers. And then we can proceed to our experiment. I will show you an example of a study that we did using regulatory T cells. These are not published data, it's just a representative study. And we were really interested in regulatory T cells since they play a role in so many diseases. But many single markers have been associated to these cells. What is then the relationship between the different markers? And these were the markers of our interest. Over 32 markers of different categories, surface, signaling, transcription markers, leading to different functions of the cell. We then tested different protocols, and we saw that the specific protocol, the Cytofix, led the best staining for both the intracellular and cellular markers that we had. We made many of our own antibodies, metal conjugated antibodies. So then we had to validate it to assure that we're having true values. And I'm using the ICOs as an example. A way that we started was by first analyzing the literature, what has been described and compared it to our results. Secondly, a side-by-side comparison of flow cytometry and mass cytometry, and if all went well, we could proceed for titration. So now we are ready to look into our results. And when you're analyzing 2 parameters in a sample, then we get a plot that allows us to look at both parameters' dimensions at the same time. 3 parameters, 3 plots. But when we go to 9 parameters, we get already 36 different plots. And that's what we usually have for flow cytometry. But now in mass cytometry that we can use over 32 parameters, we get over 496 different plots. And that many plots makes it extremely difficult to analyze such high dimensionality. And therefore, there have been algorithms created to analyze the data. So the EFCS file then can be analyzed either via Cytobank or FlowJo as an example, deconvoluted by R of MATLAB, and then we choose which algorithm we would like to use, either for cluster analysis, for dimensionality, quantitative analysis, or trajectory analysis. And these are just some of the few examples of all the algorithms that we have to use to study mass cytometry. We use the visual transformation of Spearman correlation, where the darkest red indicates the higher correlation. So we have all the markers here on the Y and X axis, and the most dark red means that these markers are more likely to be expressed simultaneously. It allows us to notice that a certain pattern among the expression of different markers. And these data, together from results from other algorithms that looked into the trajectory, the development, maturation of the T-rex, we could then know when each marker was being expressed throughout the differentiation of the regulatory T cells. And here is a summary where we can see in black what is already known within the literature of the expression of the markers in each specific development stage of the regulatory T cells, and in red what we were able to add in further detail when they are expressed, but also in more detail the different differentiation phases of the regulatory T cell. One of the analysis tools that we can use is, for example, principal component analysis, which identifies parameters that vary the most within a data set. It allows a 3D visualization. However, it assumes that the data is parametric, and it represents then in linear projection, which might lead to missing out some subtle variants within the data. Another tool could be AXENS, that stands for Automatic Classification of Cellular Expression by Nonlinear Stochastic Embedding. And here we see the data organized in a 2D map. Each dot is a single cell, then here by gradient, where the center of a cluster are the cells that look the most alike. It allows the identification of unique subsets that maybe we could not see simply by an biaxial plot. So we characterized, using the principal component analysis, the regulatory T cells from birth, from cord blood to adults. And we see that in adults, we have 6 different stages of maturation. And even though those were present at birth, the biggest population was a naive population of T-regs. If we do it in an organization about actions. Here we can see how heterogeneous and diverse the regulatory population, the regulatory T cell population is, with numerous numbers of Treg cells. And even though those seem to be present in low numbers at birth, we see that the majority of cells that we get are into a homogeneous, naive population. That's diverse throughout our maturation process. We can quantify those differences from birth to adulthood as well. And we can even classify and know what are the characteristics of each specific subset. It is relevant specifically when we look in terms of a disease. We know that regulatory T cells play a pathogenic role into graft versus host disease after bone marrow transplantation. And we knew that it was usually due to the low numbers, absolute and relative low numbers of regulatory T cells. But here we see that not only is the numbers relevant, but in these patients, we also lack the heterogeneous population of Treg cells. And we can again quantify those differences. We'll now go and look into some of the key research studies that used mass cytometry within our field of cutaneous research. By using two complementary panels of mass cytometry antibodies, Nandini Sen and colleagues, they elucidated the consequences of the varicella zoster virus on the surface and signaling network of the infected T cells. They showed that the virus infection modified the surface architecture of the T cells and promoted the skin troughing phenotype, so the T cells were going more into the skin, were also activated through the T cell receptor and PI3 kinase pathways. A study that we did ourselves, we showed distinct functional capacities of the memory T cell subsets that are migrating into the skin. Effector memory T cells secreted more Th1, 17, 22 pro-inflammatory cytokines, while the central memory T cells have higher Th2 cytokine production. Another study that we did, we showed that human central memory T cells actually can play a role in the tissue immune surveillance. We showed that most central memory T cells are tropic to either the skin or to the gut. They are present at all times in non-inflamed human skin, lung, colon, cervix, and they can really play a role in the immunosurveillance of these tissues. They play potent effector and cytokine production functions and can even alone induce skin inflammation. Guo and colleagues identified 3 new subsets with the help of mass cytometry within patients with psoriasis. They identified a CD4 positive, CD3 negative lymphoid tissue inducer cell subset, TC17, and subsets of regulatory T cells. Even confirmed that the functions of these lymphoid tissue inducer cells could be related to the pathogenesis of psoriasis. In this paper from Jim Ellison's lab, they studied the cellular response to combination therapy of anti-CTLA-4 and anti-PD-L1 checkpoint blockade for melanoma. They saw that only the combination therapy or the monotherapy for the anti-CTLA-4, but not the anti-PD-L1, induced a switch from expansion of exhausted CD8 T cells to what we'd like to have, the activated effector T cells. And even recent studies have already shown the impact of mass cytometry, for example, for COVID-19 therapy. This article shows that the inflammation caused by the virus and during treatment. It shows an overreaction of the specific subsets, such as T and NK cells, that lead the inflammation. And now the researchers knew in which subsets they need to focus, specifically when we are looking into a new disease and testing new medication. I will leave you with further studying or references, and I'm happy to take any of your questions. Thank you for joining us. Enikö or Julien?
Okay. Good. So thank you very much, Tiago, for this very clear presentation, and we received some feedback from some our members to congratulate about your clear presentation. It was very informative. Thank you very much. So maybe just a quick question, because it's a very exciting techniques. You show how it could be useful for many experimental procedure, but what is the cost actually? Do you have any idea about the cost of the machine, or the cost of the reagents, or the metals to conjugate the antibody?
Yeah. So in one of the articles that we wrote for the Research Techniques Made Simple, we tried, and there we have many more tips, and we really tried to answer some of these questions as well. Of course, it has changed, and it really depends on the agreements that they have with different pharmaceutical in the industry. But usually the cost per analysis is something like per cell 0.005%. And so it's very low. However, only around 50% of the cells that are put in the mass cytometer are analyzed. So it means that we need to have a higher number of cells to use, and we need to have at least, usually advised, 1 million cells to be in the sample, and leading to a population that is bigger than 300 events. Other costs for probe per test, maybe it's around $1.50 to $3 per probe per test.
Okay. Enikö?
Yes, we have got some questions from the attendees, and there is a question which I'm also interested in actually. So, how would you imagine to increase the number of targets to be detected? Is that possible?
So I've worked with Cytometry since 2013, and since then was when it became really novel. And just in these last years, we've seen so many new metal isotopes that have been added to the technique. Going from 30, now we have over 50 different isotopes, and more they are used in different ways for different pathways. So I do still see a lot of chance. Actually, the Cytof of the machine, the new Helios, it's capable to detect above 130 different channels. So the technology to detect the different channels is already there, and I believe they are now creating the different isotopes or targets that, tags that we can use.
We have another question about the skin sample. So if you want to analyze some skin cells, do they need to be fresh, or frozen cells could be also used for this technique?
Yeah, so both frozen or fresh can be used. It's need to have a lot of consideration in terms of the dirt that we'll get in the sample, so it needs to be highly filtrated. Also, for mass cytometry, we need to consider that the different protocols of a variance within the sample processing will induce big differences in terms of the results that we get. Therefore, it's recommended that all the samples are analyzed at once, and we can do that, for example, for mass barcoding. I will share with you again this slide, if you don't mind, because that's a crucial point of mass cytometry. To get the same results, and that is really viable when we are comparing samples, then we need to have either all fresh or frozen, and the sample processing needs to be uniform. The inclusion of the standards, what they mean, for example, having a same sample from the same donor that we always analyze together with our other samples being tested, so we can then uniformize. They use also beads within the technique, and the barcoding, which allows them to have 20 different samples in the same single tube and being stained at the same time to avoid any variety of detection of signal.
Thank you. Maybe one last question, because now we are at the end of the session.
Yes, now we got some questions. I'm afraid the time is almost up, but maybe we could take this question about the sensitivity. Is there a lower limit to it? How is the sensitivity of the method?
Then I think the sensitivity will depend in terms, do I have also a slide? The sensitivity will be in terms of what we are looking into. Are we looking sensitive in terms of if it's a rare subset, maybe we need to first increase the numbers by doing sorting, either max or flow sorting. But the sensitivity, yeah, it's quite high, but it only analyzes usually half of the cells that we include.
Exactly, that was the question about what is the lowest percentage of cells that you could detect.
Yeah, I use sometimes thousands, but then we are looking at only one single cell subset. Usually it's recommended to do at least 1 million cells for the staining, and to use different samples at the same time. So 20 different samples in one single tube, staining at the same time.
Okay, thank you.
Thank you very much, Tiago, again, and thanks a lot for having given this first talk for this ESDR Kitchen webinar. And now
yes, thank you for this excellent presentation.
Thank you guys for the opportunity. My pleasure.
ESDR member to listen this talk and to be part of this first event. And now we let Chris to end this session.
Okay, well, thank you very much, Tiago, for opening our first foray into the ESDR Kitchen so spectacularly well. Obviously, if people have further questions, we can always answer those by email. So what's next for the culinary creativity? Well, in 2 weeks' time, at the same time of the day, we'll have Julien Newton-Bishop from Leeds in the UK and David Whiteman from Brisbane in Australia, discussing the role of sunlight in melanoma pathogenesis. Is it all due to sunlight, or are there other factors? And then 2 weeks after that, on the 5th of August, I'm afraid you're going to have to put up with me again, while I'll open the batting on the molecular cuisine and talk about my personal path of discovery in psoriasis, which has occupied, I guess, most of my career. So thank you for joining today. I hope you've enjoyed it, and we'll see you all in 2 weeks' time. Bye.